This video covers the IGCSE Biology Objective 6.1.8 on the 2025 CAIE syllabus to investigate varying environmental factors on the rate of photosynthesis. Now, when we do these types of experiments, we're going to change various environmental factors and we're going to do the same experiment each time. Because one of the products that is released from photosynthesis is oxygen gas, we can collect the oxygen gas through this type of experiment. We can either count the bubbles or we can measure a volume of gas that is produced. So the first experiment or the first factor that we can look at is light intensity. And to do this in the laboratory, we set up a situation with a lamp and we set the lamp at various distances from a plant. Now what we do is we use an aquatic plant, so that way it is submerged underwater and we can then see the bubbles that are produced from the photosynthesis that the plant is doing. Basically, we would count bubbles in this particular instance. So let's take a look. In this case, we see bubbles that are produced when the lamp is very close to the plant. So therefore, a lot of oxygen would be produced in this instance, compared to a situation where the lamp is a bit further away. And so we see less bubbles being produced, which tells us that there is less oxygen being produced in that particular experiment. Now, what we can do instead of counting bubbles is we can actually invert a measuring cylinder over the plant that is submerged under the water. And you can see we're using a funnel as well to kind of guide the oxygen gas into the measuring cylinder. So what will happen is the gas will displace the water in the measuring cylinder, and then we can see the volume of gas that is produced. This is a much more accurate way of measuring the amount of photosynthesis that is produced. So you can either count bubbles or do the inverted measuring cylinder, but certainly the measuring cylinder would be a much more accurate way. So basically what we see happen here is we would then be able to graph the various light intensities versus the rate of photosynthesis. And what we would see is as light intensity increases, the rate of photosynthesis also would increase up until a certain point. And we'll talk about the plateau of the graph in a different video. Right now we're just concerned with the initial part of the graph where the line is increasing. So the greater the light intensity, the faster the rate of photosynthesis will be. In the second experiment, what we would do is we would change carbon dioxide concentrations in the liquid that this aquatic plant would be in. Again, we would have a lamp to make sure light intensity is maintained. We obviously would not change that. What we're changing here is the concentration of sodium hydrogen carbonate that's going to provide the carbon dioxide for the plant to use in photosynthesis. And again, we would count bubbles, or we would use the inverted measuring cylinder. So we would do the same experiment with now instead changing the concentrations. So instead of changing the distance of the lamp to test for light intensity, we are now changing the concentration of the sodium hydrogen carbonate. Thereby, that is our independent variable. As a result, we would see a very similar graph to the light intensity graph, which tells us that the greater the concentration of carbon dioxide, the faster the rate of photosynthesis. So again, we would count bubbles or measure the amount of gas produced. Again, this is up until a certain point, and we'll discuss the plateau at another time. The third experiment that we can do is to look at temperature. So in this case, we would vary the temperature of the water. So the lamp is going to stay at the same distance, thereby making the light intensity a control variable. We would maintain, if we needed to, the concentration of carbon dioxide in the water by the use of sodium hydrogen carbonate. And so we would then use different temperatures of water. As you can see here, we've got 0, 20, 25, and 30. But you could basically do any temperatures that you wanted to. And again, we would count the bubbles that would be produced in photosynthesis, or we could use the inverted measuring cylinder to measure the volume of gas that is produced in photosynthesis. And so what we would see is a graph that looks like this, slightly different than the previous two graphs. So what happens is, as temperature increases up until a certain point, the rate of photosynthesis will increase. At a certain temperature, we will be reaching optimum. So there is an optimum temperature for the plant, which means that at that temperature, it would be the fastest rate of photosynthesis that is occurring. Typically, that's probably around room temperature or slightly higher than that. After that point, we see a dramatic decline in the graph. So what does this tell us? It tells us that the enzymes that are in control of photosynthesis are being denatured. So we've talked about enzymes, you know what denaturing is, we've talked about temperature and how it affects enzymes. So the same thing happens in the plant with photosynthesis. Photosynthesis is an enzyme-controlled reaction, and so the enzymes that are controlling photosynthesis have been denatured, and so that's why we see a dramatic decline in the rate of photosynthesis after a certain point, which is usually the optimum temperature of the graph.